Okay, so this course is called Introduction to Bioinformatics and Computational Biology. And you wonder, what is the difference between bioinformatics and computational biology? On the course website, I had a link. Somebody made this definition. I thought it was pretty accurate. In that blog, it says computational biology is equal to the study of biology using computational techniques. The goal is to learn new biology. Knowledge about the living system is about the science. It's really more about the biology. Whereas bioinformatics is the creation of tools, including algorithms and databases that solve problems. The goal is to build useful tools that work on biological data. It's more about engineering. But then in this course, I would say we're going to use them interchangeably, okay? Because I'll show you. I also wrote a blog many years ago about the different level of bioinformatics and computational biology. For level 0, how many of you have watched Kung Fu Panda? Remember when Kung Fu Panda went in, Master Shifu was asking, show us, level 0. I remember this is about 20 years ago. No, sorry, 10 years ago. I was teaching a course, and then some colleagues came to ask me, there's so much data already. This was before high throughput sequencing. They say, there's so many microarray data available in the public domain on the NCBI website. How do we really utilize this data to really publish good papers? So I said, okay, well, depends on what kind of questions you want to answer, right? And they say, we care about modeling kind of questions. And so I feel like, oh gosh, that's like modeling for modeling's sake. I think as either a bioinformatician or a computational biologist, you have to at least care about the biology. Even for bioinformatics, the goal is to build useful tools that work on biological data. The goal has to really serve the biology purpose, right? If I would say there are definitely people who are working on level 0 bioinformatics. They work on a problem that is kind of a little bit related, but really biologists don't really care at all. Physicians don't really care at all. They don't really know how any kind of biology or clinical efforts, and it didn't really get in the door in some sense. Okay, so in this course, we're going to teach you starting with level 1 bioinformatics. This is mostly use published tools to analyze data and generate hypotheses for experimentalists. Very often, you would have colleagues coming to you, or at least to me, and say, okay, I've generated this data. We have some ideas, but or sometimes they don't have a hypothesis. Sometimes they might have some idea. They know a lot of biology about the system, say stem cell differentiation, or how a particular oncogene drives cancer, or things like that. And then they have some system, they generated data, they say, oh, can we collaborate? We want you to help us analyze the data to make sense, make sure our experiment worked. You might write some programs, but your goal is really just to help your collaborator, and they usually just give you unpublished data. You might use some of other public data to help you, but in some sense, your collaborator owns the problem. They come up with the system, they come up with the initial data, and they come to you, right? So our goal is to really help our collaborator. So that's kind of the level 1 bioinformatics. I think this is a really good point for most of the students in this course to come in, to learn the basics. What are the tools? What are the questions? How can I even make sense out of, say, even my own data for some graduate students coming to the course? So there are different level of level 1 bioinformatics. So sometimes your collaborator comes and they generate all the data. And at the end, you look at the paper, it's like all the data is in the top, and then some analysis at the bottom, and then that's it. It's the end. Or maybe they already have the hypothesis. They know exactly what type of figures they need. They just need you to help them generate. So, look at this paper. Just help me draw this figure from that paper, right? So that's kind of the lower level. The harder one is maybe they come with with some idea, but they don't really know whether the hypothesis is right. And you might be able to say, actually, your hypothesis is wrong. The data is telling us some other way. And they say, really? And then you use some other public data to help them show you are right, or whatever, you come up with new hypothesis. And then they actually listen to you. And then they use this hypothesis to design the next experiment, and then maybe generate some other high throughput data. And then they will come back to you again. You analyze, you say, okay, yes, which way is correct, and you iterate. And so I think a very good kind of collaboration is you see at the end of a paper whether the informatics and the experiments go in cycles to help each other. And so every figure has some experiments, have some analysis, and then the next experiment and that next analysis, you can see how the team really collaborate to refine their hypothesis. That would be a better kind of level 1 bioinformatics, right? So level 2 bioinformatics, I would say it's more like bioinformatics. The goal is more engineering or developing statistical algorithms, right? We want to develop either an algorithm or database for data analysis, say on a new data technique, some new sequencing technique. There's some seq every day. Early day there's RNA-seq, DNA sequencing, ChIP-seq, and now it's like there are at least 100 different X sequencing. There's like some whatever, site-seq or attack-seq, all these new things. And when the new data comes, the initial the technique developer may or may not be as savvy in terms of developing the algorithm. And so they say your collaborator comes say, oh, I'm trying this new technique. They don't even know whether their experiment worked or not, right? And there's no publicly available algorithm to analyze this. And so you can help your collaborator, but at the same time think, if I develop an algorithm, I might be able to help hundreds of other scientists. So that's why level 2 is better than level 1, because you don't just help your own collaborator, you help many other people. So you develop an algorithm and deal with the noise in the data, uncertainty, tell your collaborator whether their experiment is working, and like a general hypothesis, you'll be kind of a generally usable, and that tool you can publish on your own that hundreds of other scientists can use. And this is usually when there's a new technology under development, there's no tools available. But once this tool become quite popular, the technology become very popular, we very often also develop databases to collect all the, say, RNA-seq data, or all the ChIP-seq data, or all the CRISPR screen data. You build a database. This way, you can do integration, and other people can kind of, instead of for a biologist to go to different paper to pull the data, you can make this to be useful for a lot of other people. And so that's on the data integration and reuse. So in some sense, as a computational biologist, when there's a new technique we're interested, we develop an algorithm. But then when there are enough data for that type, we will develop databases, but also we will develop algorithm for how to reuse this data well. And then there's level 3 bioinformatics. This is more like a computational biology now. If there are enough public data available, you might be able to either use published algorithm, or you use your own algorithm that you develop in level 2, or you can even develop a new thing. But the goal is not just to develop a tool, the goal is now to make a biological discovery. So we are kind of like a biologist now, since there's so much data, right? Because very often when we have an experimental collaborator, they have difficulty even to analyze their own data, let alone all the public data available. But then if there are like hundreds of labs generating attack-seq data, like looking at all of them, you might be able to make better discoveries, right? That's more interesting. Then we kind of, now we own the problem, right? In level 1, your collaborator, they come, they have a hypothesis, they own the problem. But in level 3, in some sense, we own the problem and we make discoveries depending on the specific project. Some of them you can self-validate, but other ones you might need to collaborate with another experimental biologist to help you validate. And we are actually seeing these cases now. Very often the two groups have collaborated a lot, and the computational group kind of help the experimental groups analyze their data, and in the process, they develop tools. And then if they use the tool to also get enough public data to generate a new hypothesis on a discovery, then the experimental group help them validate. Then you really have a very, very good like a system. And so that's kind of level 3 bioinformatics. There are also level X bioinformatics. These are usually big consortium from like Human Genome Project, like ENCODE, like TCGA. So scientists from either around the nation or around the world work together. They generate a lot of data. Then they have to kind of do an integrated paper together. So in this course, I would say we should just focus mostly on level 1. We'll teach you to write some algorithms. I wouldn't say it's an algorithm. We'll teach you to write some scripts to manipulate your own data. But if you're interested, you can look at the source code for a lot of the tools that we use, and you can get a taste of what level 2 is for. There are cases recently where you see people who haven't done a tool before, they can use published tools and public data to generate a hypothesis. So they can go from level 1 to level 3 directly, which is really great. I think for physician scientists, you know, if you're pre-med, you want to go to become an MD, maybe it's asking you too much to see patients and develop algorithm, but at least you can use public tools and public data to generate some hypothesis, right? So that's also kind of happening. So bioinformatics and computational biology, I would say this is a very interdisciplinary area. The 3 disciplines, statistics, this course is offered as a statistics course, but you can see from the makeup of the student, we have people from other engineering background, biology background. I would say it's really kind of at the intersection between statistics, biology, and computer science. And interestingly, these 3 are in different importance over time. When you are a student or when you are a trainee, can you guess which discipline is more important? It's the computer science. You need to be able to manipulate the data fast enough, right? So your hands being very quick, because most of the time, say your advisor tell you, can you help me generate this figure? If you are spending all your time debugging at the end of the semester, I'm still debugging, then you really don't get a chance, right? Or if say we have 3 hypotheses, we don't know which one is right. And so you try to pull data from other sources to kind of prune out those irrelevant ones, or even from your own data. Do we see this phenomenon or this phenomenon? You have to write a little code to see which of your hypotheses is right. I would say even for people like me, I would say 90% of my hypotheses are wrong. So whoever, like if people can kind of write the code and prune out those irrelevant ones, you will hopefully reach the correct one and validate with enough public data you can publish faster. Because a lot of these discoveries or the tool development, if you're slow to that, other people have already published, right? And so initially as a trainee, computer science is the most important. Later on, and then I would say biology is probably important. You have to know what questions to ask. For example, I haven't coded for 10 years. So at the end of the semester, you will know better than me how to do this homework. So for us, we need to know what is the biological question, right? Roughly what the data have, what kind of things we can do with public data. So understanding biology will become more and more important, right? So I can just ask students to do the tool development. So over time, biology become more important. But I think in recent years, because of this machine learning, I would say there's statistics machine learning as well, and also CS machine learning. Later on, I think the statistics and machine learning will become even more important. They are not just coding, they're like real learning part. You might be able to find using this neural network or other machine learning method, you might be able to find things that biologists never knew before. And then I think the machine learning or statistics could become more important. Okay, so over time, you can see the change of emphasis over time. But for this course, I would say probably computer science is the most important, then the biology, and the third, the statistics. Okay, so even though it's offered as a statistics course, it's a very applied science. Over the years, we have students from freshmen to postdocs taking the course, and it's hard to say which one is better. It's like, yeah, you know, Bill Gates never got a Harvard. Well, he got a Harvard degree at the end, the honorary degree, right? He can still write a very good code, right? So in some sense, for bioinformatics, your ability to play with data, that's the important thing for this course. So yeah, it will be a very useful training for a lot of people. For people with biology background, I would say when there is this new X sequencing technology coming out and your advisor is so excited and says, okay, we're gonna do this experiment. It's very, very often that this student or postdoc spend 3 to 6 months just to trying to get that first experiment working, right? It's very common. And you go out and say, oh, I'm a molecular biologist. I know how to do this new XC. Oh, it's great. You're very attractive on the market. But really, think of bioinformatics as just another technique. And if you can learn it in 3 months, it's worth learning this day and night just to do this well. Of course, the more you do this, the better you are at it, especially on how to develop your own tool or how to use public data. But at least to make sense out of your own data is a very, very good skill set. I would say nowadays, for any biologist, you should learn how to do computational biology. It's a must. For statisticians or computer scientists, it's more interest. You know, it's kind of cool, really. When we were little, we were in elementary school, we heard that the 21st century is the biology century. I never believed in this, even though I ended up majored in biology. But I think in recent years, I'm like, wow, yeah, it is really happening. 21st century is a biology century. And you can see the field of bioinformatics and computational biology really move with technology development. There are still a lot of unsolved problems on microarrays, but the field has moved on because it's not as powerful a technology as high-throughput sequencing. So now people develop all the tools on sequencing. And maybe there's a new technology, then there will be new tools developed. And so is this really this course for me? As we mentioned, as you start learning the level 1 bioinformatics, your computer science, your hands-on skills, not the deep learning, machine learning stuff, is your ability to program, to manipulate data, to convert the columns and rows in the correct format to input into an algorithm, to do the README, to get it running on the Linux machine. That's the kind of very, very important. So I know a lot of you guys are very interested in learning the course, but if you have no programming before, this is really not for you. Unless you're willing to work for about 40 to 50 hours per homework. So, which is not for the lighthearted. We are gonna teach you R and Python in this course, but I would think that you at least need to have some basic background. Otherwise, this is gonna be too much for you. For biology background, I would say if you know some molecular biology, it will help a lot. I never thought this was too high a bar for Harvard undergraduate student, but maybe for the statistics graduate student, this could be tricky. Learn the basic molecular biology and the genomics. I think that will be very, very useful. The good thing is there are so many things online now on YouTube. You can learn a lot of these things. And then for statistics, you know, you need to know hypothesis testing. You need to know the major tests, you know, like t-test, you know, f-test, you know, chi-square test, you know, things like that. And also the distributions, normal distribution, uniform distribution, and things like this. And also have good intuition. I think a lot of times you don't need to have a statistical degree, but at least you need to have good number sense. So what we're gonna really teach you in this course are the major questions and the major solutions in computational biology, especially related to high-throughput sequencing. And we'll teach you the hands-on skills for data analysis and a little bit of programming, not quite as publishing an algorithm or database, but more for kind of making sense and helping you prove hypotheses. This course is totally not for statistical theory or deduction. If you came here for this, sorry, you come to the wrong place, okay? So you probably saw some brochures around this building and other campus buildings. the course is divided into 3 modules. The first part, we're going to teach you about gene expression, expression microarrays, RNA sequencing, and the single-cell RNA sequencing, and how to do analysis on those and algorithms on there. In the lecture, we will teach you mostly about the ideas behind the algorithm, what are the questions. And then in the labs, they will teach you how to run this program, how to do a bit coding. And in the homework, you will be asked to either write some programs or given the data to analyze, and you generate some result. Yeah, so that's. Then the module 2 is about gene regulation. We understand, so this one is kind of descriptive. You just know which genes are expressed. And the second part is about why is this gene expressed at this level in disease or in development, what's happening when you treat the cell with a drug, to understand gene regulation. And there's like transcription, epigenetic regulation. And also we used to cover a bit of GWAS, but we realized there are enough campus courses on epidemiology and GWAS that you can go to learn. So we kind of took that out. We'll just teach people how to use epigenetics to interpret the GWAS result. This field is really fast moving, so some of these lectures we might change from year to year. And the third one, I apologize, this is a very kind of in the third part is very cancer focused because I work at Dana-Farber Cancer Institute, and cancer is a big problem, and that's a lot of the new technologies and new data are coming to. I would say it's kind of the most cutting edge in terms of technology. And so there's also a lot of interesting data that's publicly available. And so we will talk about some major cancer project and also CRISPR. This CRISPR is genome editing, but we're looking at genetic screens, drug screens, and also on recent development in cancer immunology and cancer immunotherapy. Okay, so there are a lot of tool discussion in module 1, some biology in here, and the third part, hopefully you will be able to use the tools you learned in the first 2 modules to actually do some real exploration on data in module 3. Okay. Yeah, so is this really a good course for you? This is from the 2002 Nobel laureate Sidney Brenner. He says, all biology is becoming computational, much the same way it has become molecular. So in the 1980s, molecular biology becomes really, really hot. If you don't, because it's just so powerful, if you don't use molecular biology, you can't do a lot of the important discoveries. And nowadays, if you don't know computational biology, you will fail to make a lot of important discoveries. In fact, what is genomics without computational biology, right? Because if you don't know computational biology, if you don't know this, otherwise it's like a low input, which means you're going to do a high throughput experiment that you don't think ahead of time. You just have a lot of money and you generate a lot of data. So high throughput, you got all this data, and then there's no output. You don't know what you're supposed to find from this result, right? So I think the course should be helpful for a lot of people. Questions?